Good morning, I am Madhurima Datta, a Zoology and Biology educational content creator. Now's content for B.Sc and M.Sc Zoology Honours, Microbiology Honours, Physiology Honours and Botany Honours, paper Cell Biology, and exam for GATE exam, CSI and Life Sciences exam, CUT PG exam, DBT exam, IIT JAM exam. And Unit 1, Organelle of the Cells, and today I will discuss about the nucleus. For all of B.Sc and M.Sc in Life Sciences, please subscribe my YouTube channel Biogenesis by Madhurima and follow my Facebook account Madhurima Datta. What is the nucleus? The cell nucleus is double membrane bound organelle that serves as repository of genetic information and the master control center of gene expression, DNA replication and ribosome biogenesis in eukaryotic cells. Next, structural components. First, nuclear envelope and link complex. First part is double membrane system. Consists of an outer nuclear membrane that is ONM and continuous with the rough endoplasmic reticulum and inner nuclear membrane containing specific membrane protein that is LBR, LAP2 and aberrin. Next, perinuclear space. The 20 to 40 nanometer lumen separating the INM and ONM continuous with the UR lumen. Next, nuclear lamina. A meshwork of intermediate filaments that is lamins A, B and C underlying the INM. Next, function provides mechanical structural stability, anchors chromatin at the nuclear periphery that is lamina associated domains or LADS and regulates DNA replication and transcription. Next, LINC complex or that is also called linker of nucleoskeleton and cytoskeleton. It is composed of SUN domain protein that is INM and KASH means KASH domain proteins that is ONM. Function mechanotransduction that is transmitting mechanical forces directly from the cytoskeleton that is actin microtubules into the nucleoskeleton. Next, nuclear pore complex that is NPC and transport machinery. Architectural symmetry. Near equals to 120 molecular Dalton macromolecular assembly exhibiting 8-fold radial symmetry composed of near equals to 30 distinct proteins called nucleoporin that is also called NUPs. And structural domains that is cytoplasmic ring and filaments dock incoming cargo complex to its import receptors. Next, central scaffold or spoke complex that is surrounded the central aqueous channel near equals to 50 nanometer in diameter. Next, nuclear basket that is suspended on the nucleoplasmic site implicated in mRNA export quality control. Next, selectivity barrier. Rich in FG NUPs that is phenylalanine, F denotes for phenylalanine and G denotes for glycine repeats that forms a phase separated gel-like barrier prohibiting passive diffusion of molecules greater than 40 kilodalton. Next, transport mechanism mediated by karyopherins that is importin or exportin driven by the RAN-GTP or RAN-GDP gradient maintained by RAN-GEF and RAN-GAP that is cytoplasmic pump. Nucleolus and subnuclear bodies. The nucleus is highly compartmentalized despite lacking internal membrane barriers. Liquid-liquid phase separation that is LLPS drives the formation of non-membrane bound nuclear bodies. Next, this chart is very important for the CSI net that is subnuclear domain, key molecular marks and primary function. First is nucleolus. Domain name FC, DFC and GC. Key molecular markers are fibrillary, nucleolin and UBF. Primary function tripartite site for rDNA transcription that is FC and pre-rRNA processing that is DFC and ribosome submit assembly that is GC region. Next, nuclear speckles. It is a molecular markers are SC35, SR splicing factor and primary function are storage and assembly domains for pre-mRNA splicing machinery. Next, causal body subnuclear domain. Key molecular markers cholins, SMN protein and primary function are biogenesis and modification of snRNPs and telomerase RNA assembly. Next, PML bodies. Key molecular markers are PML protein and SUMO1. Primary functions are transcriptional regulation, DNA damage response, apoptosis, and viral defense. Chromatin architecture and spatial organization. First is higher order packaging. Nucleosome core, that is 147 pp DNA wrapped around an octamer, that is 2 into H2A, H2B, H3, H4, and chromatin fibers organized into topologically associating domains, that is stats, stabilized by CTCF and cohesion complex. Next, chromatin domains. Euchromatin, transcriptionally active low density region enriched in histone acetylation, that is H3K4Me3. Next, heterochromatin, transcriptionally silent condensed region divided into constitutive telomeric enriched in HK9-methyl-3 and facultative, that is developmentally regulated enriched in H3K27M3. Next, chromosome territories. Individual chromosomes occupy discrete non-overlapping spatial domains within the nucleoplasm rather than forming a random entropic tangle. Next, nuclear matrix and laminopathies. Nuclear matrix or nucleoskeleton, a non-chromatin proteinaceous network remaining after chromatin extraction, that is primary composed of lamins and matrins and SAF A or B, serving as the anchorage site of replication factories and RNA processing complex. Pathological relevance or laminopathies. Mutations in LMNA gene, that is encoding laminin A and C, causes a range of genetic disorder due to mechanical fragility or altered chromosome tethering. First is Hutchinson-Gilford progeria syndrome or HGPS, that is single nucleotide mutation creates a cryptic splice site, then truncated laminin A, and that permanently retains of farnesyl group, and then nuclear envelope, blebbing, premature again, and loss of heterochromatin. Next, Emery Dreyfus muscular dystrophy, structural defect in INM architecture and affecting the muscle mechanotransduction. Now the topic has ended.